So, hello. Um, there once again. We had a little bit of trouble here trying to get the screen up for you all. Um, but anyways, I'm going to just discuss a few things here. This is just one of the many topics that you probably already saw in my previous introductory course as we discuss a little bit of everything in the 4 hours time crunch, or in the 4 hours, that we had um, during that discussion. So, in this one, we're going to mainly review on HIV medications. However, I'd like to also talk about some other important concepts because there's a lot of drug interactions that involve with some of these medications as well. I will be right back. What? I'm gonna log these doors. No one's coming in. But I want to use my computer. So I'm just going to give a couple more minutes for people to join this review. Um, Hello, can anyone hear me? Um, I'm just testing to see if you guys are able to hear me or not. Give a few more minutes so you guys can um, join. So, um, I'm just going to go over it now. Um, Looks like we've already, um, started in the previous presentation. We talked about like the different types of disease states and the medications, some small things about each medication, um, and I also I will talk a little bit about those today as well, but it's not going to be like comprehensive like the previous lecture. Um, this one's just mainly going to focus on the antiviral drugs. Specifically, when we talk about antiviral drugs, um, when a host cell is infected by a um, HIV virus, you would think that it's going to actually start by first attaching that virus cell to the actual human cell, and then it's going to go infect it and then replicate. So before it gets there, it needs to go through a couple of steps. It needs to first fuse. It needs to first enter. They need to go through and gather all those proteins, and then it needs to reverse transcribe. So you would typically see DNA going to RNA, but in this case, RNA is actually going to DNA. So it's going to do reverse transcriptase here, and it's going to program it so the cell's going to be infected. And so what happens here is it's going to then replicate itself uh, around the body, and then it's going to, infect the overall system. So, excuse me. So, sorry about that. So, as I mentioned, um, like when it comes to HIV medications, there's different types of classes or um, of medications, and those medications act on different um, parts. So you probably you may see this, but like you might see some of the classes like NRTIs or NNRTIs, PIs, and its specific role in terms of creating that type of infection by the HIV virus. And then um, you have some drugs that act at the part where it kind of fuses and enters into the system. So those kind of inhibitors are involved to prevent that from happening. So it's important to understand that as well. So let's take a look here. Um, I don't know why it's happening like this. It seems like um, we have had a little bit issues, here and there. But anyways, I'm going to quickly go over these mechanism of actions of various HIV medications. So at least you have the understanding of that concept. Um, if you already had the time, Previously, I posted a NABP NAPLEX outline. Um, I would recommend taking a look at those slides and kind of looking through and seeing what types of um, things to look out for when looking at these drugs and its treatment as well. Um, I'll list out a few pertinent side effects and some interesting points and some shortcuts along the way so you guys can see all of that. Um, so, if you've already reviewed my previous lectures, um, you can see here that there's some other topics that's discussed in terms of like infectious diseases. So, like antibiotics, pneumonia, UTIs, skin and soft tissue infections, intra-abdominal infections, the GI infections. You might see osteomyelitis and then endocarditis, sepsis. Um, and we kind of already briefly talked about vaccination in the previous lecture, which I think is it's very important to know. Um, and then there's some other special cases. These are things like, how to, um, help patients in terms of like renal adjustments and stuff. We'll be looking at all of those things as well. But today, specifically focusing on HIV and its medications and the mechanisms of those drugs. I think it's going to be very valuable, especially since it's one of the hardest topics some of you might find in terms of studying for like any sort of pharmacy exam, including for the NAPLEX. So we'll look at some different approaches here so you guys can see a little bit differently of how I kind of laid it out here. So it would make it a little bit easier for you to kind of understand, learn the material um, while reviewing your materials as well. So let's take a look. Um, so see here, this is a, let me try to, what happened here? Okay, first of all, are you able to see the slide? Anybody able to see the slides here? And if not, then I can post another link for you guys to rejoin if it's not, if you're not able to see it, because it looks like you only see that one slide right now, unless, okay, I think you guys should be able to see it. Um, so you can see here some of the steps that occur, like when it comes to HIV infections, like first it needs to attach to the host cell, bind to the CD4 receptor. So there's a receptor on the host cell that's the CD4 and it needs to bind there, and then it will start fusing with its the envelope, the membrane envelope of that virus to the actual host cell before it can release all the programming. It's kind of like you can think of it like a virus, like a computer virus. Before the system was working normally, but now we're injecting like a Trojan horse virus or some kind of virus into a system. So that virus is going to kind of take over every process. So essentially, that's the same thing as what's happening here. It's like a virus is being injected right into the human host cell. And then that virus is going to go ahead and program itself definitely to the DNA. So the RNA is going to reverse transcribe to DNA and then infect the host DNA and create a new viral RNA. And the viral RNA is going to definitely um, go through a next step here. And you can see here, number 5, that the RNA is used as a genomic RNA to make viral proteins. So RNA is typically where the proteins are made anyways. If you've heard of like cell biology or in a class lecture and when you were in pharmacy school or pre-pharmacy, you probably already know that you need RNA to make those polypeptides and proteins. So in the way the virus is making its proteins or um, to make its replication and assembly and then start maturing. So all of that things to strengthen the virus. So it's kind of a very simple way to kind of look at it. I wouldn't worry too much on the infection as much as how to treat it. I would understand how to treat each and every part of this infection. So each of these steps represent how the infection occurs. But now we're going to look at the treatment mechanisms and where these medications target in terms of um, preventing the infection or slowing down infection. So let's take a look. So in this next slide here, um, you can see here, like I listed one of the classes of medication, which is the NRTIs. Now that includes things like tenofovir, emtricitabine, lamivudine, abacavir, and you can see here also that with that particular medication, there is um, a pharmacogenomic um, test that is used, and that's an important one to know for the test, the HLA-B 5701. So understand that for abacavir. Um, so what do these medications do? First, and I put these notes up here as well for your information so you can read through them. You're going to see like a fake nucleoside is incorporated into the DNA. And so basically it's going to program it so now the DNA cannot like elongate. And then it will result in chain termination and block the reverse transcriptase. So it's going to work right there in the reverse transcriptase. I kind of drew this diagram out for you so you can see it. This one right here is kind of a representation of like, and I'm going to kind of draw it out here for your reference. But you can see here, this one is kind of like a representation of like the enzyme because, transcriptase, enzyme. So, so kind of like the enzyme, kind of like making a reading of this reverse transcriptase from RNA to DNA. So like RNA to DNA. And you can see here, this is the viral RNA. Viral RNA. Oh, and this was like the human DNA. So let's look back at this diagram here real quick. See what's cool about this is that you could see here, um, this is actually occurring right here at this step here, the reverse transcriptase. And so you could see here, that's actually in like the third step here, it's occurring right here. And this is where like um, the pre-integration complexes, and then like you can see here how it's following through the viral RNA is right there. You see that reverse transcriptase and enzyme here is also inside this uh, I guess you could say it's like an envelope or like a bucket of, materials that you're going to be using to assemble the whole product. So, anyways, so what it's going to do is it's going to block this part. It's not going to um, make it work properly, because what happens is DNA is going to get into this and the transcriptase. So it's essentially going to work like that. And let me give you a little bit better visual here so you can see it. Wanted to make it a little bit more clear so you can see it. Um, just give me 1 second. So just give me 1 minute, okay? Hold on. What do you need? Yeah, you can. It's fine. Yeah, that's fine. Yeah, I'm not on the recording right now, so it's fine. indoors. Hi, sorry about that. Um, had to interrupt you for a bit here, but as I was going through this, yes, it's going to infect human host DNA by implementing some of the viral RNA proteins that are the genes and programming into the DNA and making it a viral, um, making it a viral DNA. So what's happening here is that the reverse transcriptase is going to definitely be interrupted, and like it's going to happen right from that point of view. So you can see here, these are some of the medications. This one you have to specifically test for the HLA-B 5701. So just don't forget about that for Abacavir. And I kind of pointing out like how it's kind of inhibiting that reverse transcriptase enzyme, so it doesn't affect that um, DNA of the human. So you can kind of see that in this diagram here. The next one I wanted to talk about was the NNRTIs. So similar kind of process. It's going to stop the reverse transcriptase. Both of them block the conversion of HIV RNA to HIV DNA. So these both the NRTIs and NNRTIs going to work internally in the process of infecting the cell, but the drugs are going to be different. You could see here efavirenz, evaporine, rilpivirine, and then doravirine. You can see here the difference in our side effects here too, like specifically with the NNRTIs. Specifically with the NNRTIs, you can see here like CNS, rash as side effects. And like you can see here, like I think efavirenz has some other specific side effect. I would recommend kind of looking into that. Specifically with efavirenz, there is a pertinent side effect there, especially things like vivid dreams and nightmares. Now, I don't know if we're going to get a test question on this, but that is one important pertinent side effect I would kind of remember with the efavirenz. As I remembered, I think I've seen it somewhere, so I recommend you at least kind of know that. But these are some of the drugs listed here. And so what is NNRTI? Does anybody know what NNRTI and NRTI stands for? Does anybody know? So NRTI is nucleotide reverse transcriptase inhibitors, and NNRTI is non-nucleotide reverse transcriptase inhibitor. So which one's going to bind directly to the reverse transcriptase? NNRTI or NRTIs? So it's important to know this in terms of mechanism is that the NNRTI is going to bind directly to the reverse transcriptase. Now you could see here in the diagram, it's kind of showing both of these, but specifically the NNRTIs bind directly to the reverse transcriptase. Um, but the NRTIs do play some role in the reverse transcriptase inhibitor, but it's going to cause DNA chain termination. So the end result's pretty much going to be different. This one's going to prevent the enzyme inhibition. The other one's going to prevent the DNA chain, prevent that elongation of the DNA chain. So at least understand that main difference between the NRTIs and NNRTIs, just for your reference. Okay. The next one here is usually like the booster drugs, like the PIs, which is the protease inhibitors. It's going to prevent that new HIV from becoming immature HIV. So you could see here, like the protease, that's an important component. It's kind of like a little important, like a heart inside, like the molecule, the HIV, and kind of like preventing that from being implemented there. So it's going to block that HIV from becoming more mature. And when it becomes immature and non-infectious, that's what we want. We don't want it to be infectious. So it's going to prevent the cleavage of one of the polyproteins, the GAG polyprotein. So these are some key things to understand when it comes to mechanism of action of HIV medications. You need to have a familiarity with NRTI and NNRTI, and don't be confused with these two. Even though they both deal with reverse transcriptase, the end results can be different. And also with NRTIs, they actually mimic the nucleotides. So whereas non-nucleoside do not mimic nucleotide. So you can think of non as non-nucleotide mimics, and NRTI is nucleotide mimics kind of thing. Hopefully that kind of helps. So you can see here, like the protease inhibitors kind of like stopping this HIV from becoming more mature. So protease kind of like another enzyme for, making the proteins to do its work inside the viral cell. So it's going to need this so it can become a full virus. So without it, it's going to be useless. So that's pretty much the point of the protease inhibitors. Then the next one here is the integrase transfer inhibitors. Integration, you're integrating something, you're going to block the integration of that RNA or HIV DNA into the CD4 cell DNA. So you can see here, okay, so reverse transcriptase, reverse transcribe RNA to a DNA, became viral DNA. Viral DNA is now going to insert its block into the actual DNA within the nucleus, and that's where the integrase inhibitor is going to play a role in it's stopping that integration. So this picture is really, really good. It actually kind of points a few things out for you, so at least you can get a idea as to the processes here. Dolutegravir, bictegravir. You could see here in the name tegravir is here. Tegravir, integrating. I hope that kind of helps with integrase strand transfer. So just understand that. This next one is going to be a little bit confusing to some of you, but like entry and fusion inhibitors. Take a look at that, how it's kind of the HIV virus is actually infusing itself into the human host. You can see here, and that's when you're going to use something like fuviritide, which is a fusion inhibitor to prevent the fusion of that envelope to the human host cell. So that attachment's going to fail because it's not going to allow the HIV virus from getting into the human cell and infecting its genetic parts. So, and then also the other one is the entry inhibitor to take a look there in the Mavorok here. Mavorok is interesting because it binds to this. There is another receptor, the CCR5 receptor, so it's going to kind of be an antagonist to the CCR5, so the virus can't bind to that part. And so HIV cannot attach and enter the cell, and it only works if virus is CCR5 tropic. If it's not, then this medication is not going to really work. So just kind of, wanted to let you know, have an understanding of that. Kind of drew it out here, but all three of these diagrams kind of, hopefully they kind of help kind of see the big picture here of the entry and fusion inhibitors. And then here, finally, since we already completed most of these classes or all of these classes here, this is my little special chart that I made using the previous shorts, showing a breakdown of some of the mnemonics you could use while studying for the exam. You can see here like NRTIs, kind of used each letter from the first name as tildes, kind of like thinking terminate, and then NNRTIs like rand or like roads ending. So like remember I talked about chain termination and the enzyme inhibition. So previously you saw that how the NRTIs cause chain termination and NRTIs cause enzyme inhibition. So I wrote N kind of thinking about like, well, the enzyme's not going to do any more work in creating that virus because the road's going to end there. PIs, was just like lard. Also think of things like AVIR here. You see AVIR, AVIR, AVIR at the end. So those are like PIs here. And one specific one I want to talk about was ritonavir. Let me see if I can go back here. Where's it? There is probably one thing about ritonavir that is important. It's one of the strongest CYP3A4 inhibitors, so it's kind of an important point to understand. Speaking of inhibitors, going back to the previous lecture when I talked about clopidogrel and omeprazole, I did not mention this, but clopidogrel is actually a prodrug. So if the omeprazole is going to inhibit that CYP2C19 of the clopidogrel, you can see here it's going to actually prevent it from being activated and working in the body. So I wanted to just point that out from yesterday, um, just so you know. Uh, and then you have other drug like cobicistat as well, which is a PK inhibitor, um, stat or CStat. There isn't really any acronym I could come up with. If you know of any, you can just, you know, make one up to remember. Think lacking a road driver. So I guess this one's referring mostly to um, uncontrollably kind of driving through it. So, but PIs generally you can just think of it from like the last three letters without the G, because the G will make GRA, because you can see those are the integration, in, integrate strand trans, you know what I mean, the integrase inhibitors. So um, that one's pretty easy to remember. You probably don't need an acronym for that one, but just in case, I listed it here for you. This last one was just a snippet from the short here, the entry inhibitors. I actually like the one where it said enfuvirtide, like fusion. You could say fusion, FU in the enfuvirtide name, so more like fusion, and the other one's like rock. So it's like you can probably make something out there. Um, So imagine it blocking the HIV tide from entering. That's another way to think about the Mavorok and the Enfural tide. Um, and in the initial combo treatments, so some of these treatments have a combo in them, and I'm pretty sure some of you may have seen these in the pharmacy one way or another, like Biktarvy, for example. I know I have, so I've seen it being dispensed in the pharmacy. Um, it is not a cheap drug, but it is sometimes used when dispensing it. So, um, big Tegriver, you can see here in the name now. If I had to give you a question on this, or if the test gives you a question, they're probably going to ask you what medications these combos are made up of. And so I'm going to give you a quick quiz here if any of you guys are interested. Um, want you to see if you can remember without cheating or looking at the diagram what classes each of these combos of Biktarvy is made up of. And think to yourself, do I know these classes of medication like Biktarvy? What classes of HIV medications do you think make up um, this combo? So, like Taggart, which of these classes of medications is associated with bictegravir? Does anybody know? No? Um, so good to see here, bictegravir, more like integrate strength um, inhibitors. So you could see here the INSTI is used here in this case. Then you got here, the next one, emtricitabine. Emtricitabine. Remember the word teals? So, think that as emtricitabine in there. And then, I didn't even mention the NNRTIs as well have the word VIR within the name there. So you can see here, it's not at the end, it's in the middle here. So it will kind of help you understand the differences between NNRTIs and NRTIs. So, so check it out. So see here, like how it's kind of listed here within the name, like not at the end, within. So that will kind of, help you understand the differences between these drugs here, in case you get a question on a test. So the next one, emtricitabine, that's going to be NRTIs. Then tenofovir, and as you recall that tenofovir is another drug as an NRTI as well. Remember the word teals. Okay. And then the next one is Trumet. Does anybody know the classes of medications associated with Trumet? So Trumet is going to be dolutegravir. So it's going to be integrase in D right here. Then you got apicavir. That's going to be a TEALS as well. So that's going to be NRTIs. And then you see here lamivudine. Okay, so what did we talk about here? TEALS again. So you got two NRTIs and you got an integrase inhibitor as well, the INSTI. So that kind of, at least you can see how to make up some of the classes of these medications in a combo drug. And then the next one here, Dovato. It looks like there's an INSTI here, dolutegravir, and then lamivudine, which you can see here is a NRTI. Okay. I think you guys pretty much have an idea as to kind of naming some of these HIV medications. Notice here in the PIs, it has an AVIR at the end. So if you see that in the name, that's a PI. These are the NRTIs, that NNRTIs. NNRTIs has that VIR in the name within the middle of the drug name, so you can kind of make it out that way. So yeah, that's pretty much all I could tell you. Just know these. Kind of like understand the mechanism. Also, what is one thing about abacavir that's different? Remember I talked about a genetic test, HLB 5701, remember? So definitely make that note so you guys can review it. So I think, I hope that all of this was helpful. Take note of some of these shortcuts as you look at the names of many of these HIV medications. I have a few questions here in the next slide so you guys can kind of use it as a review. If you guys remember all the drug names or not. So definitely, if you want to kind of peek back at these, you can. You can write out the acronyms. You can, use this as a guide. Just at least have an understanding of these types of medication. Some of the side effects, important side effects, like the one I just mentioned here about the enfuvirtide, um, that one, or efavirenz, which is definitely one that kind of has an association with kind of vivid dreams and stuff. Um, Also, rash and CNS type of side effects, particularly when it comes to NNRTIs. Um, and then the nucleotide reverse transcriptase inhibitors, that one seems a little bit different as well. Um, and knowing the differences and how they work, that that one is going to prevent the elongation of the chain, the other one's going to prevent the um, enzyme inhibition. So you know that difference. PIs, your priori still have protease that plays an important role in maturity of the HIV virus. Um, so PIs can prevent that, and ritonavir being like a CYP3A4 strong inhibitor. So being unable to understand the drug interactions, many of the CYP3A4 drugs interact. So pharmacogenomics play an important role in this whole chapter as well. Typically with PIs when it comes to CYP3A4 inhibitors. So you would probably see a lot of drug interactions with it. Um, and actually could be used in a good way because sometimes it can help kind of boost the other medication as well if it needs to be used a little bit more. So it's good or bad. They got the entry and fusion inhibitors, and you saw how like that can kind of prevent like the envelope from binding or fusing to the human host cell membrane. Then you saw the integration strand inhibitors. Um, you probably saw them in many of the combos here as well. So just take a minute and just review these things. Fusion inhibitors, especially PK boosters, um, combos, just kind of look through that. I think the PK boosters is the one I was talking about, the ritonavir being a PK booster, but you can also see it as a protease inhibitor as well. So it plays a role in both parts. So do not get confused between that on the exam. Does anybody have any questions? That's pretty much it in this lecture. Wasn't a very, I didn't want to make it too long for you, so. But we will cover more detailed information in the future posts, just to keep you updated with knowledge and stuff. So, anyways, thank you for watching. Have a good day. Bye.